Okay. Hello? Yeah. Okay. Hello, so welcome to this first live session on this course, The Physics of Biological Systems. I hope you people have been watching the lectures and you're enjoying the lectures and hopefully you're learning some new and interesting things. So before people come online, so let me address a few issues which have come up in the forum many times. So one of the persistent issues has been that people are asking for textbooks. I understand that, of course, it would be very helpful to have a book ready, but unfortunately, as we have posted on the forum, the main books are mostly copyrighted, so therefore we cannot really provide you with a PDF or an electronic copy of them. What I do try to do is that I try to upload my lecture notes as much as possible, and I've uploaded the lecture notes till this week 4, or I think maybe some even from week 5, and I'll continue to do that going forward. If there are aspects that you would wish me to elaborate upon in some more detailed notes, you can please post on the forum, and I'll try to do the best that I can. So the other thing is that, of course, in a few, I think before the end of this month, so by the end of February, we'll also have like a detailed sort of transcript of what I'm saying in the lectures and whatever we are covering in the slides. So you'll have access to that set of notes as well if you wish to, go over the things that have been talked about in the lectures as well. Something else that somebody posted in the forum is that whether some papers might be available, and that is indeed a very good idea. So I'll try to do that both for the weeks that have already been posted, so weeks 1 through 4, as well as the coming weeks. So as and when I see that some concept which we have discussed during the lectures, it would be helpful to have some additional paper, some research papers if you wish to read. So I will upload those as well. It may be current papers, it may be slightly older papers, but papers which are sort of landmark papers in the field, as it were. So I'll try to upload some of those papers. The thing is that, of course, it's somewhat difficult because these are going to be research papers. They might be somewhat difficult to sort of read through in full detail, but at least if you read a little bit of it, if you get the idea or the sense of what they're trying to do, that in itself would be, I think, a good lesson from the papers. So please feel free to type any questions that you may have, and I'll try my best to answer them. Now, there was one question that was already posted in the forum, so I thought I would address that. So I think this was by someone called Sri Krishna, and he was asking about this, the diffusion limited regime, so the case of perfect absorbers. So we did the case of spherical absorbers, where we found out the diffusion limited flux, which was 4 pi dAc naught. And then I did not derive, but I talked about some other geometries like the disk geometry and the ellipsoidal geometry. So the question was that when I wrote down the result for the ellipsoidal geometry, I wrote down this dn dt, the rate at which particles come to this surface of this ellipsoidal cell, as 4 pi dAc naught divided by log of b by a, or 2a by b, I think, where a is the semi-major axis and b are the two semi-minor axes. So it's true, it's a spheroid, it's not really a general ellipsoid. So in a general ellipsoid I would have 3 axes, a, b and c. Here I take that the two smaller ones, b is equal to c. And so I had that formula, and as Sri Krishna very rightly pointed out, that if I were to take the limit that A goes to B, I should recover this the result for a sphere, which is just 4 pi dA C naught. So that's a very valid question. So the thing is that this result that I wrote down, that was written in a certain limit, which I omitted to mention. So thank you for asking this question. It was written in the limit when A is much larger than B. Okay, so it's if you imagine, it's like a cigar sort of a shape. The ellipsoid or the spheroid, the ellipsoid of revolution, is like a very elongated cigar. So the semi-major axis A is much larger than the semi-minor axis B. In that limit, if you were to solve the boundary value problem, the diffusion equation or the Laplace equation, you could show that your analytical results would reduce to that factor log 2a, by the denominator would be log 2a over b. Of course, if you were to then try to go to the limiting case of a sphere, you could not use that result anymore, because that result is derived using the assumption that a is much larger than b. Okay, so thank you for asking that. So it's that result that I wrote down is true in that limiting case of a cigar-like geometry. All right. So the other thing that that was posted was to, let me see, what was the other query that was posted? Was about this application of biological physics in research and in industry. Right. So, of course, biological physics is a very active field right now. In fact, very recently, last week or so, the National Academy of Sciences in the USA designated biophysics as one of the major subfields in physics. So it's going to undertake a survey which is called a decadal survey, which it does every 10 years in order to figure out where the field of physics as a whole is headed. And this time, for the first time, in fact, biological physics has been included in this decadal survey, because increasingly people realize that this is one of the areas of the future. More and more research is being done in this area of biological physics in trying to model biologic data from these biology experiments in a very quantitative way in order to build up, in order to build an understanding of the physical principles involved, right? So I'll give you a couple of examples that are mostly drawn from my own research, because that's sort of the thing that I know best. So one of the problems, for example, that we study is how sort of embryos develop, starting from this single cell, right, into this complex organisms like human beings or whatever other organisms that you're talking about. And at these very early stages, so of course this is a very complex phenomenon, right? In some sense, this is a question of how life emerges. And it's a very complex phenomenon with lots of chemical processes, biological processes, but additionally also lots of physical processes. So there are these molecules. Molecules need to get transported from one end to another. So there are diffusion processes going on. So we've talked about diffusion in these first few weeks. So there is a lot of diffusion going on. There's a lot of active transport going on, in which cells actually spend energy in the form of ATP to push things from one place to another, from wherever they have been produced to wherever they need to go. So a lot of these processes ultimately involve physics concepts, of course, how this interplay of these chemical reactions in these spatially confined domains take place in order to give rise to this developmental body plan, in order to give rise to organs and so on. So this is a field known as developmental biology, and increasingly, there is a lot of physics modeling that is going on in there in order to trying to make sense of the data that is emerging out of the experiments. And we'll talk a little bit about about how developmental biology, how patterns form and how organs form and tissues form, briefly towards the end of the course, towards the last couple of lectures maybe. Then another problem that I'm sort of interested in is about how these how the chromosomes, all of our cells, inside our cells we have a nucleus, and inside the nucleus we have our DNA, right? All of us know about DNA, and this DNA is sort of organized into chromosomes, and these chromosomes organize themselves inside the nucleus in a certain way. And if one thinks about it, of course, there is the sequence of the DNA, the A, T, C, G, so on, but there is also a physical problem as to how you pack these sort of long strings of DNA into this very tiny volume inside the nucleus. So you must, balance energy, you must balance entropy, and you must find out a recipe for how the cell sort of organizes this complex chromosomes in such a way that it can go and read the information. It knows exactly where each segment is, and it can read that information with a great deal of accuracy. Okay. So this is just two sort of random examples where a lot of physics concepts come into play. There are, of course, multiple other examples that one could talk about. I just chose these two to give you a brief idea. So, as I said, I'll of course continue to, what I'll do is that I'll upload papers as we go along from these topics that we've been talking about. So, diffusion, and then low Reynolds number physics, so swimming of microorganisms in low Reynolds number. These are not really part of required reading for the course, I would say, but for those of you who are interested in some further reading, you can read up, you can look at the papers that you're interested in and just read up on them a little more. The other question that was posted was about these applications to industry, and of course there one of the major fields that I can think of is in this research and development in medicine, for example, drug delivery. And there, you have to design sort of drugs that will go to the right place and to will attach to the right chemical components and do exactly the right function that you want to do in order to combat whatever disease that you're trying to do. So it involves, before you can sort of test it on humans or animals even, it involves a lot of development, and a lot of the development is sort of computational. So you have these complex models that take into account all of these forces that we'll be talking about throughout the course. Sort of interaction potentials, how hydrodynamic forces play a role in this delivery, what sort of shape should it be, how should it interact with these molecules of interest. So all of this requires knowing a lot of the physics that is involved in these molecules, the physics and the chemistry. So that's of course one major place in industry where things like this can play a role. Then also in devices, so for example, you want to perform experiments in some bacteria, and you want to isolate maybe one bacterium species versus another. You can often use these hydrodynamic forces, as we are talking about in these when we're doing the Navier-Stokes equations, in order to build the devices to separate different strains of bacteria and so on. Right. So one more question, if I remember correctly, was on the application of quantum mechanics in biology. Now I'll be a little careful with this question. So it's not something that we'll cover really in this course. That's because mostly when we talk of biological systems, we are interested in a length scale that is of the order of microns, so micrometers, And quantum effects really become important if you are at smaller length scales of the order of maybe nanometers or angstroms even. Okay, so when we are talking of slightly larger systems, so then the quantum mechanical effects do not really play a major role. You can treat them classically, like we've been doing and like we'll continue to do, and still understand a fair bit of this physical biology. Where it does become important, of course, is, as I said, in the nanometer scales, And so you can imagine that where it will become important is these single molecule sort of studies. So if you're looking at properties of single molecules, how they go from maybe one structure, one conformational state to another conformational state, their quantum mechanics can become important. For one obvious example that I can think of is in these vision receptors in our eye. So, you know, when a photon strikes these receptors, there is a sort of structural change from one conformation to another. So the absorption of the photon is, of course, a quantum mechanical phenomenon, and that drives the structural conformational change. And that conformational change gets interpreted back. The signal induced by this conformational change gets interpreted back in the brain, and ultimately results in you seeing objects, whatever caused the photon to fall on your eye. Another obvious example is, of course, the most basic example that I can think of, which is photosynthesis. There again, you absorb a photon, and you store this you store this energy, and you convert it into chemical energy to be used later for the whole business of life. But the level, so of course there is, if you're talking about the single molecule sort of level at this nanometer or angstrom resolution, so you have this field of quantum biology where you must take into account the quantum mechanical effects. However, at the levels that we are interested in, so we are in this course, we'll mostly be talking about processes on the level of a cell or maybe subcell, but not really a lot of single molecule physics. So at these levels, when you're talking in the scale of microns, hundreds of nanometers or microns and so on, the quantum fluctuations are really not that important. The temperatures are, of course, room temperature. So another limit when... So the quantum effects are... another limit where it is important is, of course, at very low temperatures, and that is not really a relevant limit for biology, because biological systems, us, organisms, living organisms, plants, and so on, all live at around room temperature thereabouts. So in that sense as well, the quantum effects are not really very important. So there is a field of quantum biology involving single molecules. But that is not really something that we'll be looking or focusing at much in this course. We'll be dealing more with the classical physics, and even within the realm of classical physics, there are these whole variety of unsolved problems in biology that we as physicists or as engineers with our training in mathematics, can help biologists in understanding what drives these systems. Okay, so I think in case there are no further questions, I'll stop this live session. We can, as the course progresses, we can of course have more of these. So, and in between, of course, please continue posting your queries on the forum, and we'll try our best to answer them. And after maybe some time has passed, if there is a need, we can do another live session where you can feel free to sort of ask any questions or any doubts. Okay. All right. So I think that'll be all for today. And please continue. I hope you enjoy your lectures and you continue to learn new things through the lectures. Okay. Thank you.